Hey folks, this is lesson 5 in our acids, bases, and salts unit. I did skip over lesson 4 for this because I do want to cover that with you in class. So the lesson you're going to work on today is on indicators that tell you roughly the pH range of a particular solution. So we're going to skip over this do now question because that refers back to lesson 4. So an indicator is a compound, and this particular compound is called Congo red, and it changes color, or it is a particular color at a particular pH level, and it can change color if the pH of the solution changes in one direction or another. So this particular indicator, Congo red, is a definite blue from a pH of 0 to a pH of about 3.5. And then you see this transition color between blue and red is this purplish color. And then it tends to be red only from a pH of 4.5 and up. No, this one says it changes from 3 to 5, but you know what I'm saying. So it's a different color because the molecular structure changes with exposure to acids or bases. So just remember, the pH of a solution can indicate how acidic or how basic something is. Again, we will go over the more nuanced meaning of pH in class on Thursday, but this is just a chart that shows you a variety of solutions and what their pH levels are, whether they fall into the acidic range or the basic range. You also did that, investigated this in that simulation. So an indicator is a compound that changes color when pH crosses a certain range. So it'll be one color at certain pH levels and a different color at other pH levels. It basically changes color when a hydrogen is added or removed from the molecular structure. So if you think of it this way, here's the hydrogen and here's the rest of the formula for the indicator. It can separate into its separate components. So it would be one color with the hydrogen attached, and it would be a different color with the hydrogen detached. So you have a reference table M that gives a list of 6 different indicators. There are lots, there's dozens of different compounds that can be indicators, but this is just 6 that you would need to know how to interpret on this reference table. So the way that this works, let's look at this methyl orange, for example. It gives you the approximate range of pHs when the substance does change color, and it gives you the 2 different color options that you have. So the way that you read this is, if I have a solution, do you hear my children yelling? It's wonderful. If you have a solution that has, let's say, a pH of 2.5, and you add a couple of drops of methyl orange to it, the solution will turn red, because at any pH less than 3.1, the compound is red in color. Now, conversely, if you have a solution that has a pH, let's say, of 5, and you add a couple of drops of methyl orange to it, then that solution would turn yellow, because methyl orange turns yellow in any pH that is higher than 4.4. Anywhere in between here, the compound would be a blending of the colors involved. So it would be orange in color. Think about your mixing of primary colors, right? Red and yellow make orange. So the way that, again, you read this chart is that if the pH of the solution you're testing is less than the number that's on the left, the solution will be the color that is on the left. If the pH of the solution is higher than the number on the right, the indicator will turn the color that's listed on the right. So the in-between for phenolphthalein would just be a really, really, really faint pink color. It's completely clear in anything less than 8, and it's like a bright fuchsia color in anything higher than 9. So if you're in between 8 and 9, it would be very, very, very faint. We don't really concern ourselves with the in-between colors. We just look at what is it, at pH lower than the left of the range, and what color is it in a pH higher than the right side of the range. So we're going to look at some example questions here. So this is a Regents question. Which solution will change red litmus to blue? So all we care about is this final color. Okay, so we focus on litmus, and we want litmus to be blue. Litmus is blue at any pH level that is higher than 8.3. So you have to think to yourself, okay, well, what types of solutions have pHs that are higher than 8.3? And those would be bases. So this question, really, you just have to find the base. So you can go to reference table L, where you have a short list of bases, and you would see sodium hydroxide listed there. So that's the correct answer. Choice 1 is an acid. Choice 2 is a salt, so that's not going to do anything. And choice 3 is actually an alcohol. And I'll go over with you in class how do you distinguish between an alcohol which has OHs and then a base which has OHs. But I'll review that with you in class. So look at this one. Which solution will turn phenolphthalein pink? So we focus on just the phenolphthalein, just this color, and this number. So anything higher than this number, phenolphthalein will be pink. So if the pH is higher than 9, again, we are working with a base. So we can eliminate choice 1 because choice 1 is an acid. Choice 2 is also an acid. That they're both on table K. That is carbonic acid, that second one. Choice 4 is an alcohol. Again, I will review how to distinguish that. So our answer here is choice 3. When you have a metal with hydroxide, you can be sure that you have a base. So here's a word question. A blue solution containing an indicator was tested with a pH meter and it was found to have a pH of 5.5. Which indicator could this be? Okay, so just looking at our scenario here, we have a solution that is blue, so it has some substance in it as well as an indicator. And then they tested with like a digital pH meter and it came up with a 5.5. So we want to pick an indicator that is blue at a pH of 5.5. So first of all, let's eliminate the ones that do not have blue as an option, because you would be surprised at how many people would be like, oh, it's clearly phenolphthalein. If blue is not an option, you cannot pick blue, okay? So now let's look at our options here. So bromethymol blue has the possibility of being blue, but only if the pH is higher than 7.6. So that has to be eliminated. Litmus has the possibility of being blue, but only if the pH is higher than 8.3. So that's not a possibility. Bromcresol green is blue at any pH higher than 5.4, and thymol blue is blue at any pH higher than 9.6. So since we're looking for a pH of 5.5 and blue, our answer would have to be bromcresol green. So that solution, whatever it was with a pH of higher than 5.5, has bromcresol green in it. That is giving us that blue color. Sometimes you need to use more than one indicator to kind of narrow down your pH range. So going back to this example, if I tested a solution with bromcresol green and it turned blue, all I know is that and I didn't do a pH meter, I just tested it with an indicator of bromcresol green and it turned blue. All I know is that the pH is higher than 5.4. There's a lot of things that have a pH higher than 5.4. Pure water has a pH higher than 5.4. It's 7. I would gladly take some. Thank you, delicious. You know what else has a pH of higher than 5.4? Bleach. You know? So sometimes using just one indicator and seeing the color of it isn't going to tell us enough information. So that's what we're looking at here, is how can we use different indicators in various samples to kind of narrow down our pH range. So this scenario, in bromcresol green, a sample of solution turns blue. So again, like before, we know the pH is higher than 5.5, but like, would you go swimming in that? I don't know, because a 7, an 8, a 6, absolutely. A 12, probably not, gonna get a little irritated here. So what you would do is you would take some more of the solution in question and you would test it with a different indicator. Okay, so we took another sample and we tested it with bromethymol blue and it turned yellow. So here's bromethymol blue. The yellow color will show up at any pH that is less than 6.0. So now we have a range of pH. We have a pH that is greater than 5.5 and it is less than 6.0. that is a much narrower range for us than saying, oh, it's just higher than 5.5. It could go all the way up to 14. But now we've really narrowed it down and we know we're dealing with a weak acid. It's slightly less than 7, but not crazy. Okay, so that's how you'd use multiple indicators. So let's take a look at this one with multiple indicators. A solution turns yellow with thymol blue and blue with bromethymol blue. So we take each one individually. So thymol blue and yellow means that the pH is, we know the pH is less than 8.0. Okay, so again, it could be 7, it could be 6, it could be 2. So we'll test it again. We're gonna take another sample and test it with bromethymol blue, and it turns blue. Bromethymol blue indicator is blue at pHs that are higher than 7.6. So now we know we're dealing with a range between 7.6 and 8.0 for our solution, which would make it a rather weak base because it is still quite close to 7. Other indicator that we use sometimes is called universal indicator. So we have like vials of pH paper that can turn multiple colors. That paper is soaked in universal indicator. So you get a variety of colors depending on the pH of the solution you're testing. In actual situations involving indicators, I've mentioned hydrangeas to you. So if you got a pack of hydrangeas and you plant the seeds and you wait for these shrubs to grow and you split a pack with a friend, and you notice that you might have blue flowers on your plants, but your friend might have bright pink flowers on their plant, even though they came from the same packet of seeds. It's because hydrangea flowers, the compounds that give them their pigments, are sensitive to pH of the soil. So if you have hydrangeas in your yard and they bloom this spring and summer and you see you have blue flowers, that means you have soil that is lower than a 5.5, so it's quite acidic. If you have purple flowers, you're 5.5 to 6.5, and you have pink flowers, you are higher than 6.5 of your soil. There are a lot of reasons why pH is important in agriculture. So you probably learned in living environment that enzymes in your body require specific pHs for them to work properly. Well, plants have enzymes too. So different plants, like let's say foods, all these are like flowers and foods, they grow best at different pH levels, probably because the enzymes that help their metabolism are sensitive to pH. So if you go, especially this time of year, if you go to a garden center or Home Depot or Lowe's and you look at the different fertilizers that are available, there are fertilizers that are specific for certain types of plants, depending on, if you want like larger fruits, extra fruits. So something like tomatoes, for example, prefer soil that's quite neutral, right? It's pretty close to 7. So you can buy fertilizers that will help to regulate the pH of your soil, depending on what you're growing in that part of your garden. So this is just like a pretty comprehensive list of various flowers and fruits and vegetables. Other reasons, I guess you could say, for the importance of pH and indicators are called extremophiles, which are organisms that live in extremely acidic or extremely alkaline environments. Heliobacter pylori is a bacteria species that lives in your digestive tract normally. And it, since your stomach is extremely acidic, you have hydrochloric acid in there, it has the ability to survive as part of the symbiotic relationship in your body at that very low pH. It does attack the stomach lining. If you have an overgrowth of Heliobacter, normally it doesn't do much, especially if the mucus lining of your stomach is adequate. But if you have like a depletion in the mucus lining of your stomach, the Heliobacter can attack the lining of your stomach and cause ulcers in your stomach and your small intestine. This diagram is showing you how CO2 can acidify the soil, acidify the water and change the pH of oceans, and that can lead obviously to environmental concerns. And when the pH of the water environments drop, animals have to, it increases the solubility of the calcium carbonate that makes up their shells. So things like clams and oysters and scallops and mussels and coral and starfish and urchins and all these different organisms produce calcium carbonate that make up the structure of their shells. And if the oceans become more acidic, those shells structures start to dissolve. So the animals have to work harder to replenish that and kind of waste energy. Oh, look, it is red cabbage. That is also an indicator. Oh, and this is just different foods and the different colors that they'll be at various pH levels. And that is it. So I have some practice for you guys to work on with indicators, and I will see you soon. Bye.